Hi everyone, I'm Hyun-Myung, a PhD student from Texas A&M University. Today, I'd like to talk about one of our recent works concerning optimizing the performance of high-throughput virtual screening pipelines. This study has been done with professors and researchers at Texas A&M University, Brookhaven National Lab, and Rutgers University. Professor Byung-Jun is my advisor. My presentation will proceed in the following order. First, I'd like to explain the motivation of this study. In the next chapter, I will provide a high-level overview of the proposed methods. Then, I will present some of the simulation results. Finally, I will conclude my presentation with some concluding remarks. Effective selection of the potential molecular candidates that meet certain conditions in an immense search space has been one of the major concerns in many real-world biochemistry applications, in particular in drug discovery. For example, since the coronavirus disease 2019 outbreak, an urgent scientific mission has been established to find molecules that can proceed to later stages of the drug design protocol against the COVID-19. In such a task, there are two fundamental challenges. Above all, the number of drug molecules is enormous. Second, accurate evaluation of the molecules is fairly expensive in general. Therefore, accurate and efficient selection of the potential drug candidates from a huge set of drug molecules has been the key factor determining the success of the screening problem. As a result, the need for an efficient high-throughput screening pipeline arises frequently in various fields beyond drug discovery, including biology, chemistry, and materials science. This figure shows the impeccable pipeline, which consists of multiple deep learning-based surrogate models for docking and orthodox GPU, coarse and fine-grained binding-free energies. To date, Impeccable has screened over 4.2 billion molecules against over a dozen drug targets in SARS-CoV-2. It has already led to the identification and experimental validation of over 10,000 compounds, resulting in over 40 hits that are progressing to advanced testing. However, to date, there have been no systematic policy to manage such HTVS pipelines. Although there are subtle variations, the stages in the HTVS pipeline have been mainly used to sort the incoming samples and pass them to the next stage sequentially. So, we were curious if we can optimize the performance of the HTVS pipelines in terms of computational cost and throughput. In this work, we proposed two general optimization frameworks to optimize the performance of the HTVS pipelines. The first framework applies to the HTVS pipelines when there is a computational budget constraint. The second framework jointly optimizes the throughput and the computational cost. In other words, it maximizes the throughput while minimizing the computational cost. Here, I'd like to provide a very high-level overview of the proposed methods due to the time constraint. Please check our manuscript on arXiv for more details. We first generalized the HTVS pipeline problem. Let me explain how the general pipeline works. We have initial search space X. Stage S1 evaluates all the samples in X through F1 and pass them whose score Y1 is equal to or greater than lambda 1. The shifted samples are evaluated in stage 2 via F2 and filtered out some of them whose score Y2 is less than lambda 2 and so forth. As a result, the number of samples gradually decreases based on the stages. Here, our main objective is to maximize the throughput with proper consideration of the computational budget. This is done by optimizing the screening thresholds lambda 1 to lambda n-1. Note that we assume that the screening threshold of the last stage lambda n is given by experts. This assumption is reasonable, because usually such Htvs pipelines have been used to minimize the number of samples for accurate experimental evaluation involving human experiments, which is more than often much more expensive. The key idea is to estimate joint distribution Fs of scores y1 to yn computed at different stages of the pipeline. To this aim, we may consider diverse existing spectral estimation techniques. In this study, we assume that joint score distribution follows the multivariate joint Gaussian distribution and utilize maximum likelihood estimation or expectation maximization algorithm depending on the availability of the label. Based on Fs, the screening thresholds are optimized to maximize the throughput while minimizing the computational costs. The first framework applies to the screening problem under a computational budget constraint. Again, I'd like not to go into details. Basically, we optimized the screening threshold from lambda 1 to lambda n-1 by maximizing the reward function R based on the estimated joint score distribution with the budget constraint. If there's no computational budget constraint, but still we want to minimize the computational cost, we can jointly optimize the efficiency and throughput at the same time. To this aim, we normalized the reward function and the computational cost function such that the maximum value of each function is equal to 1. Besides, we introduced alpha that balances between efficiency and throughput. We found that 0.5 showed fairly balanced performance between the throughput and efficiency. We performed the simulations to validate the performance of the proposed optimization schemes based on both synthetic and real-world pipeline data. However, due to the time limitation, we present simulation result for real-world example only. We considered the long non-coding RNA screening problem because they are closely related to hard-to-treat diseases such as Alzheimer's disease, cardiovascular disease, and several cancers. To this aim, we formed a high-throughput virtual screening pipeline for screening long non-coding RNAs by integrating various long non-coding RNA prediction algorithms with different accuracy and computational costs. We collected nucleotide sequences of RNA transcripts on the human reference chromosomes consisting of 48,752 LNC RNA sequences and 106,143 protein coding sequences from gene code version 38. We filtered out the sequences containing invalid characters other than A, U, C, or G, and the sequences whose length exceeds 3,000 nucleotide. Lastly, we applied a clustering algorithm CD-HIT to LNC RNAs and protein coding RNAs respectively to remove redundant sequences. As a result, we have 140,733 RNA sequences consisting of 39,785 LNC RNAs and 64,948 protein coding sequences. We selected 4 state-of-the-art computational algorithms estimating the coding potential probability: CPC2, CPAT, PLEG, LNC Finder. To better understand the algorithms, we first evaluated the performance of the algorithms on the pre-processed dataset. As shown in the left table, LNC Finder outperformed all the algorithms in terms of accuracy and sensitivity at the cost of the highest computational complexity. CPAT was the second best performer in terms of accuracy and sensitivity and recorded the best specificity. CPC2 and PLEG were the third and worst performers in terms of accuracy, sensitivity, and specificity, respectively. CPC2 and CPAT were not only computationally efficient but also highly accurate compared to the worst performer plaque. Based on the evaluation result, we set the LNC finder as the final stage and constructed a basic HTVS pipeline structure as shown in the upper left figure. Besides, we computed Pearson's correlation coefficient between scores estimated by the algorithms. CPAT showed 0.93 of the correlation coefficient to the LNC finder, yielding the highest correlation among the prior algorithms. We estimated the joint score distribution Fs via the EM algorithm with 4% of the samples. In addition, we assumed that the lambda 4 is equal to 0.2. This figure shows the performance comparison results of the optimized HTVS pipelines with various structures. The X and the Y axis stand for the computational budget and the number of potential candidates that pass the last stage, respectively. The black horizontal and vertical dashed line represent the number of true potential candidates and the computational cost for the complete evaluation of all the samples based on only the last stage LNC finder, respectively. The black vertical dotted lines are located at intervals of 1/10 of the original cost. Underneath each dotted line, the number of potential candidates selected by each optimized pipeline is summarized. The complexity of the initial stage in the pipeline determined the point where the pipeline starts to select the potential candidates, and the correlation between a stage and the last stage was closely related to the slope of the performance curve. The pipeline, including the second stage S2, associated with CPAT that is highly correlated to the last stage, showed the steepest slope of the performance curves. As a result, all the optimized pipelines, including CPAT, regardless of the order of the stages, identified from 45,697 to 47,466 of the potential candidates out of 50,266 true candidates at only 50% of the original cost. Interestingly, the proposed framework was effective to robustly configure the pipeline such that it consistently performs well even in the presence of the stage that is computationally complex but less correlated to the last stage. Specifically, although the optimized pipeline S1, S2, S4 outperformed the optimized pipeline S1, S2, S3, S4, the performance gap was relatively small. Considering that HTVS pipelines can include various types of platforms, some of which may not be closely correlated to the last stage, to screen samples from completely different perspectives, the proposed framework optimized the HTVS pipeline in a robust way. We validated the performance of the pipelines jointly optimized through the proposed framework when there is no budget constraint. On average, the optimized pipelines selected 48,372 potential candidates out of 50,266 true potential candidates throughout the simulation at the effective cost of 3,067. As we expected, optimized pipelines, including the second stage S2 associated with CPAT, achieved relatively high computational savings compared to those without S2. The optimized framework was indeed effective to reduce the performance degradation caused by the presence of the stage associated with a less correlated and computationally complex algorithm plaque. In other words, the selection capability of the pipeline optimized via the proposed framework was mainly dependent on the best performing prior stage in the pipeline. Furthermore, the proposed framework significantly reduced the performance dependency on the order of the stages within the pipeline. For example, when one utilizes all the stages, the optimized pipelines selected 48,402 potential candidates on average and consistently saved the computational resources from 42.27% to 44%. These are some concluding remarks. We presented two computational frameworks optimizing the performance of the Htvs pipelines involving surrogate models with different complexity. The key idea is to estimate the joint distribution of scores computed at different stages of the pipeline, based on which the screening thresholds are optimized to maximize the throughput while minimizing the computational costs. We proposed two frameworks. We first considered the case where the computational budget is fixed and the goal is to maximize the throughput within the given budget. Next, we considered the case where we aim to maximize the throughput of the Htvs pipeline while minimizing the overall computational costs at the same time. We demonstrated the performance of the proposed optimization schemes based on both synthetic and real-world pipeline data. We formed a high-throughput virtual screening pipeline for screening long non-coding RNAs by integrating various LNC prediction algorithms with different accuracy and computational costs. We showed that our proposed optimization frameworks can lead to the significant computational savings at identical or comparable screening throughput accuracy. Thank you for watching my presentation. Please email me or leave comments if you may have any suggestions or questions. I will be happy to answer. The manuscript is available on arXiv, so please take a look if you are interested.